All right, this narrated PowerPoint video is going to finish Chapter 4, where we're working on membrane transport. So specifically, this video is going to cover bulk transport. That's covered in section 4.13. And bulk transport is when vesicles or vacuoles are used to move large amounts of substances across membranes. Let me repeat that. I don't have this as a note on here. Bulk transport. Bulk means a whole bunch. Like when we have curbside pickup at our homes and the city comes by and picks up your old sofa or refrigerator, that's bulk pickup. Bulk trash. So bulk transport is when a vesicle or a vacuole is used to move a large amounts of substance across a membrane. So the first kind of bulk transport that we're going over here is called endocytosis. So endocytosis is when substances are brought inside cells using vesicles or vacuoles. And endo means inside. So endocytosis means movement to the inside of a cell. That's what it means. So it's using vesicles or vacuoles to bring substances into a cell. And the figure, which makes up a lot of the slides space, is showing you three different kinds of endocytosis. They're doing different things, different jobs. The first one is called phago or phagocytosis. Phago or phago means to eat. So, for example, one image or example that might stick in your head is if you've ever seen a dog eat poop, feces, that's called coprophagy. Copro means feces and phagy means to eat. So coprophagy is eating shit. So phagocytosis is loosely translated to cell eating, because look at the figure on the far left. What's happening is a cell is wrapping its plasma membrane around some kind of target, like a big chunk of food, maybe an entire cell, like a bacterium. And the plasma membrane wraps around that large object and brings it inside in a big, gigantic bubble of membrane called a food vacuole. Once that's brought into the cell, then I can ask you, which organelle is a eukaryotic cell going to then use to digest that material in that food vacuole? The answer would be lysosomes. Lysosomes would be sent to that food vacuole to digest what's in there. Look at the bottom left of the figure. It's showing you an electron microscope image of an amoeba engulfing a bacterium and using phagocytosis to bring that bacterium inside. Now the middle panel is showing you what's called penocytosis, and this is loosely translated to cell drinking. So what happens here is the plasma membrane has little indentations in it, little cups in it. And those little cups, those little indentations fill up with fluid on the outside of the cell. And then those little bubbles pinch off inside the cell, and those are vesicles. And so those vesicles are just like gulping or drinking in whatever solutes, whatever materials are outside the cell drinking, penocytosis, forming vesicles to bring substances in. Take a look at the electron microscope image in the middle panel. It's showing you a cell lining a small blood vessel, and that cell is bringing in lots of fluids by penocytosis. So it's a very rapid way to bring lots of stuff into the cell at once. Finally, receptor-mediated endocytosis is the most complex, really. So you've got these little cups in the plasma membrane. They're called coated pits. Coated pits. So they're a pit, like a little cup in the membrane, and they are coated with receptors and very specific molecules. We call those ligands. Very specific molecules attached to those receptors. And once those receptors are all filled up with those target molecules, then the coated pit closes off and forms a vesicle and brings those targeted molecules inside. So whereas penocytosis gulps in whatever is outside the cell, just non-discriminately, receptor-mediated endocytosis is being very specific and targeting certain molecules to bring them inside with vesicles. So the very bottom right figure is showing you a coated pit that is being filled up with specific substances and then forming a vesicle on the inside of the cell. So those are the three types of endocytosis. All three of them are bringing substances into cells using vesicles or vacuoles. Phagocytosis is like cell eating, forming a vacuole. Penocytosis is cell drinking, gulping in fluid in vesicles. And receptor-mediated endocytosis is targeting specific molecules with receptors in coated pits and bringing them inside in vesicles. So we just went over what endocytosis is. What about exocytosis? Exo means outside. And so exocytosis is moving, a cell is moving substances to the outside using vesicles or vacuoles. Vesicles are tiny, vacuoles are big. Exocytosis, like the outside of a spider or an insect, is called the exoskeleton, the skeleton that's on the outside. Exo, outside. So this is the method that eukaryotic cells use to release or expel large amounts of substances to the outside of a cell. So what's happening? Take a look at the figure. It's very instructive to understand what that picture is showing you. Here comes a vesicle, okay, in the picture, in the bottom left. Maybe that vesicle is coming from the Golgi complex. And this vesicle contains a bunch of substances that need to be released to the outside of the cell, maybe to make the extracellular matrix, maybe to release digestive enzymes, maybe to release hormones. There's a number of different reasons why a vesicle might be going to the plasma membrane. So that vesicle fuses to the plasma membrane. The phospholipids that make up that vesicle just flow right into the plasma membrane after it sort of crashes into it. And the vesicle basically turns inside out, and the substances are released to the outside environment. Take a look at the transmission electron microscope image on the right-hand side of that figure. It's pretty awesome. It's showing exocytosis. It's showing a vesicle caught in action as it's releasing substances to the outside of the cell. So hormones, neurotransmitters, enzymes, etc., they're made in the endoplasmic reticulum. Review. They're sent to the Golgi. Then they're sent to the plasma membrane and vesicles to be secreted by exocytosis. Now, contractile vacuoles, I mentioned those a little bit earlier in the last part about osmosis and osmoregulation. So something like a paramecium or freshwater protist might fill up with water because the outside of the cell is hypotonic, has low solutes and high water. So water goes into that amoeba or paramecium, and if it doesn't get rid of that excess water, it's going to explode by lysis. So contractile vacuoles are filled up with water and they go to the plasma membrane and turn inside out and release water to the outside, bailing water to the outside. So these are examples of exocytosis, basically the opposite of endocytosis. Both are using vesicles and vacuoles to move substances across plasma membranes. I'm hoping this is working, that you're able to get linked to my YouTube video that I made with a whiteboard to show you exo and endocytosis. So watch the video, take any notes that you wish to before moving to the next slide, but I'm showing you the kinds of things that I just talked about, but just in a more dynamic kind of way by making drawings on a whiteboard. So take a look at that if you can, and then move on to the next slide. Okay, we're now on the last section of chapter 4, active transport. Active transport. You need to know what it means. Definition. Active transport is sort of the opposite of passive transport. Remember, passive transport means something is moving without the input of any extra energy. Like a ball rolling down a hill, that's passive transport. Or your bicycle rolling downhill, that's passive transport. Active transport means energy is required to move a substance against or up its concentration gradient. So when you're riding that bike and you want to go uphill, you're not going to be able to coast uphill. You're going to have to pedal. You're going to have to use energy and use your muscles to get up that hill. That is active transport. A car could coast down a hill, but you need gasoline, you need energy and gasoline or diesel to move that car uphill, or an electric car uphill. So, okay. So what is the source of energy for active transport? What do cells use as their energy source? ATP. You're going to learn more about ATP in the next chapter. All right, metabolism and energy chapter, and the chapter after that too. But ATP is a little energy molecule that your cells use for anything that requires energy. Anything. Your muscles, your nerves, they require immense amounts of energy to work. ATP is a little molecule that your cells produce as an energy source. So that's the energy source. And ATP supplies the energy for proteins in your plasma membranes to actively transport or move substances, to basically pump them to the other side. You need energy to move them from low concentration to high concentration against the gradient, against the direction that they naturally want to move. So the figure here is showing you this. You got a transport protein. There's the phospholipid bilayer on the left side of the figure. It's showing you there's a higher concentration of substance up on the outside of the cell on the top and a lower amount on the inside. But the cell needs to move the substance from low to high. So it takes ATP, the middle panel of the figure, is supplying energy to basically grab onto those molecules and move them from low to high. That is active transport. All right, so this slide is talking about the sodium-potassium pump. And this is probably the most important sort of active transport example that you can learn, because if you go on and take anatomy and physiology or microbiology, I guarantee you're going to need to know about the sodium-potassium pump and how it works. I guarantee you. So the sodium-potassium pump uses energy, and of course that energy is in the form of ATP. And basically this pump is moving for every 3 sodium ions that are moved out of a cell from low to high concentration to build a gradient. there are 2 potassium ions that are moved inside the cell from low to high concentration. So this pump is actively moving these 2 kinds of ions to the other side of the membrane to build their gradients. Nearly 1/3 of the energy expended by your body's cells is given over to driving these pumps. That's how important these sodium-potassium pumps are. So I've got a link here too to a video to see how it works. So finish going over this slide, taking some notes on this slide, and you can only access this video through the actual PowerPoint slides. You can't access it through this YouTube video. So if you go to my PowerPoint slides in the module and open them up, you'll be able to access the video. It comes from your textbook. So now look at the bottom of the slide. I've got some additional notes. The result of the sodium-potassium pump is to generate a concentration gradient with more sodium outside the cell than inside the cell. And cells exploit or use this gradient in some important ways. First, and you'll really get into this in A and P, the conduction of signals along nerve cells is done with the sodium-potassium pump. So really important, nerve cell conduction. And a second reason I'm giving here is the transport of important molecules into cells against their concentration gradients. So those are 2 reasons. So take a close look at the figure. It's showing you for every 3 sodiums that are getting pumped out, 2 potassiums are coming in, and it's showing you that ATP is being used as an energy source. Okay, this is still in section 414, and this is the topic of co-transport or coupled transport. Same thing. So our cell membranes, plasma membranes, have many facilitated diffusion channels for sodium. But the sodium is only transported from out of the cell to into the cell with another substance, and that is called coupled transport or co-transport. So remember from the last slide, the sodium-potassium pump. Cells use energy to pump sodium ions out of the cell to make a gradient. Once there is a gradient, more sodium outside than inside, that sodium is just itching to come back in, to diffuse in from high to low. So the point here is that there are specific proteins, transport proteins, put into your plasma membranes, and the sodium ions can move through those transport proteins from outside the cell to the inside, moving down sodium's gradient. But as the sodium moves in, the energy from the sodium moving in also helps transport other substances like sugars and amino acids into the cell at the same time. That's called coupled transport or co-transport. So it's a very common way for cells to accumulate sugars and amino acids and bring them to the inside of the cell where those sugars and amino acids are needed. Watch the video. I wish there was a figure that your textbook had that I could easily put on the slide, but there isn't. But I do have the link to the co-transport video. And again, you need to be opening up the PowerPoint slide, using PowerPoint. I've got the file in the module, and then you can get access to the video. You can also go to your textbook. And if you go to section 4.14 in your textbook, you'll have access in the e-text, the electronic text, to these videos. Okay, so that's it. There's one more slide, and let's check it out, and then we'll wrap this up. Okay, so I sure hope that you had done your reading questions for this section before you started using your worksheet and taking notes. In your reading questions, I put a blank version of this table, and asked you to fill it in using your information in your textbook. It's a fair amount of work, but it was a good kind of heads up to these different ways that substances move across membranes: simple diffusion, facilitated diffusion, exocytosis, active transport, co-transport. It's all summarized on this table. Very, very cool. It's on page 101 of your textbook. Now, I'm encouraging you to use that blank table again and to see how well you do in filling it out after going through these notes and hearing this material and so on. Because if you can fill out this blank table, then that is showing you, yourself, how much you know, how much you've learned and assimilated and so on of this material about the transport of materials across cell membranes. So it takes a lot of work to learn this stuff, no doubt. But this would be, I think, a good way for you to invest some time and see what you know and what you don't know by trying to fill out that blank table. And always, always focus then on what you discovered that you don't know. That's where you need to spend your time.